Hi, my name is Samantha Chong, and I will be presenting you my science fair project, which is about the difference between bacterial growth in personal and public areas. The pandemic has affected everyone, including shutting down schools. Since then, kids are now allowed to go back to school to get a proper education. That means that hygienic practices are now practiced more frequently. You can reduce the amount of germs on objects by having products containing alcohol and micelles. How micelles work is by surrounding dirty particles and destroying them. Some types of bacteria that can be found in schools are Staphylococcus aureus, Pseudomonas aeruginosa, and Streptococcus pneumonia. Both Pseudomonas aeruginosa and Streptococcus pneumonia can both be found by doing improper hygiene. For example, not covering your nose or mouth when sneezing or coughing. You can find Staphylococcus aureus commonly found on your nose, even if you're healthy. Bacteria can best grow in a warm, wet, and dark environment. Being in the dark will help because the ultraviolet light that comes from the sun will kill bacteria. In the warm temperature, bacteria increase in size, and in the wet environments, it will help the bacteria properly grow. Some areas that should be sanitized frequently are doorknobs, keyboards, and faucet handles. When using hand sanitizer, it is not able to remove all germs on your hands, but when using soap and water, it will. The effects of bacteria are very dangerous. You could get wound infections, bloodstream infections, and etc. Some ways you can prevent spreading bacteria is by washing your hands and covering your nose or mouth when sneezing and coughing. My hypothesis is that the more contact an area gets, the more bacteria there will be. I also predict that the doorknob sample will have the most colonies, knowing that kids will constantly touch them throughout the school day. The materials that you will need for this project is 12 strips of paraffin tape, 6 petri dishes that already have the agar solution in it, 6 cotton swabs, 6 vials in a Ziploc bag, a pile of gloves, a pair of gloves, a permanent marker, a source of water, some sort of box that will fit the petri dishes, and a piece of paper. For the methodology, first you wear gloves, so there won't be any contamination with the bacteria. Label the lids of the petri dishes A1, A2, A3, and B1, B2, B3. The A's represent public areas and the B's represent personal areas. Do that as well for the vial lids. When getting the samples, take the Q-tip out, put a little bit of water on it, and rub it on the area so it'll get all the bacteria on it. And then quickly put it in the vial to prevent contamination. Once you have all of your vials ready to put it on the petri dishes, you take the Q-tip out quick and then rub it on the agar solution in a zigzag motion. Do it evenly for all samples. And once you're done with that, quickly put the lid on and put the paraffin tape on it so it'll secure it. Wait a span of 1 week and check on it for 2 days each time to see how they grew. To count the amount of bacteria colonies that are there, you can count them by the colonies. That's how I did it. Or you can use this formula. The amount of colonies growing times 10 squared the amount of square units. And if there's too many colonies to count, get a grid paper, put it into 4 sections, and just count one section to get the most approximate amount. As you can see in the first picture, that's how I set it up when starting to rub the Q-tips on the agar solution. And in the second picture, that's how I stacked the Petri dishes in a shoebox. In the third picture, that's how I figure out the approximate amount of colonies because there's too much. And in the fourth picture, that's what the drawing looked like. Comparing the A's and the B's, the A's have a lot. For A3, it had 2,104. And for B3, it had 37. They're both the highest in their selections. And when you look left to right, you can see that they grow higher and higher because they're growing more and more colonies throughout the week. In these pictures, again, when you look left to right, you can see more and more colonies growing and they're getting bigger. That's because I put them in the right environment, a warm, dark, and moist environment. The samples from public area/objects had more bacterial growth than personal area/objects. In the first check-in, A1 had 53 colonies, A2 had 0, A3 had 1,172, B1 had 0, B2 had 21, and B3 had 0. In the second check-in, A1 had 208, A2 had 0, A3 had 1,712, B1 had 2, B2 had 30, and B3 had 4. In the third check-in, A1 had 392 colonies, A2 had 0, A3 had 2,104, B1 had 3, B2 had 37, and B3 had 5. In total, the public samples had 2,496 colonies that formed, and for the personal samples, it had 45. In conclusion, my hypothesis was supported by my experiment. The public area samples had the most colonies, with the total of 2,496. The mouse sample had the most colonies in the public area, with the amount of 2,104, and the eraser sample had the most colonies that grew in the personal area group with a total of 37. Some ways that you can help reduce spreading germs are covering your mouth when coughing, covering your nose when sneezing, and frequently washing your hands or sanitizing. I also think a reason for these results is because in public areas, a lot of people are having contact with these areas, and they may not all have the same hygienic practices, while in personal areas, the surfaces are having contact with the same people and tend to be less. I hope that this experiment will bring attention to people and how they are keeping up with their personal hygiene practices. This is a good reminder that personal hygiene is good for our health, our own health, and the health of everyone that is around us. For future changes, I would like to take samples from the same areas and compare them over a long period of time to see if there is any difference from now and when there are less restrictions. Thank you for your time, and do you have any questions?